Hi, everyone. Please take your seat. We'll be starting now. Our last lecture of the day before the lab will be given by Jacopo. Jacopo is currently doing his PhD within the group of Professor Cecilia Clementi. Over there, he's mainly working on applying machine learning technique in the development of coarse-grained models for protein, and currently simultaneously he have completed his master in physics at the University of Freie Universität Berlin. Thank you so much, Jacopo, for giving this lecture today. Let's welcome the speaker.
Thank you. Hope you can hear me well. Thank you very much for the introduction. So I'm here today to represent Cecilia Clementi's group, and today I want to give you a bit of an overview of what we are doing. This is not all my work. I just did a small piece of it. The presentation is more general and involved lots of application of coarse-graining. Today we speak a lot about coarse-graining, so some part of my presentation are already were already done by Gianni, for example. But I try to give you a sort of more general idea of coarse-graining and try to discuss some application also far from the direct molecular dynamic approach. So let me start with with this introductory idea. Why do we need coarse-graining? Well, because essentially when we have to deal with biological system, we need to study a very wide range of time and landscape, right? So we might be interested in study very detailed and small processes, like, for example, like the electronic structure. In that case, we wouldn't use very high resolution method. But for studying more a bigger system in general with longer time scale, we cannot use those kind of first principle method. All right, we need to use some more simplified model also in order to get some insight, some physical insight on the system we want to study. And this is the idea of coarse-graining. So essentially behind this approach of coarse-graining, there's the basics idea that not all the degrees of freedom are relevant. It's kind of the same that you do when you do molecular dynamics under the Born-Oppenheimer approximation, right? So you are assuming that the electronics degrees of freedom are not necessary to explicitly solve your problem, and you treat them as a sort of mean field. This is the same idea, but push a bit farther, right? So we want to be systematic. This thing was already anticipated by Gianni. And what I want to point out is that the main idea here is that we want to really be systematic in the way we do coarse-graining. So we want to fix a specific resolution. For example, we want to, I don't know, eliminate the solvent. All the solvent atom in a molecular dynamic simulation. Or we want to eliminate some structure inside the protein. So we want to retain some very fundamental elements that are associated to the protein, for example. So like carbon alpha atom only. In this case, we eliminate all the atoms inside the protein, and we only take one bead for every residue. Of course, the solvent is also eliminated. The problem at this point is, of course, well, we have this very simplified structure, but how this actually evolved, how we can describe this structure, what's the Hamiltonian for this system? And in order to answer this question, there are several approaches. So the first is the so-called top-down approach, where you model basically your coarse-grained Hamiltonian in order to match some low-resolution observable, meaning, for example, some experimental observable. Okay, you want to design a model that is able during simulation to reproduce experimental value. This is the case, for example, for Martini force field that is widely used in molecular dynamics simulation. There's also another approach that is the so-called knowledge-based approach. It works pretty well when you already, for example, know the folded structure of your protein and you want to explore some neighborhood of the folded state. So you insert inside your model some important structure that you already have. And there's also another method that is the so-called bottom-up approach. That is the one that we will discuss more in this presentation. And basically here, the idea is that you start with a very high resolution description for your system, like an explicit atomistic description, and you want to obtain a coarse-grained representation for this system, imposing some sort of thermodynamic consistency. And we will discuss this later. So this is the outline of the presentation. We will start with what Gianni mentioned before, so how we can build a coarse-grained force field for protein that is transferable in configuration space. So here the final goal will be really to prove you and convince you that we are able to extrapolate, make prediction out of the training set. Then we will discuss some interpretation methods, so how we can apply interpretation techniques to those machine learned coarse-grained model in order to get some insight of about the model prediction and in order also to validate our model. We will then discuss how we can use the coarse-grained formalism to incorporate nuclear quantum effect inside simulation, and this is something that normally is ignored, but can be very relevant in particular thermodynamic condition. And we will conclude with a very small overview of how we can quantify multibody effect using a top-down perspective in a coarse-grained model. So for the first topic, we need to understand what thermodynamic consistency is. Gianni already talked a bit about this, but I want to redefine everything just in order to be sure we are speaking the same language. So the idea when you want to do in general bottom-up coarse-graining is that you start with a high-resolution system and you map this high-resolution system to a lower resolution representation. So you need to define a mapping operator, that is this operator here, that basically takes set of atoms and map them to a single point in your CG space. You can think about it in this way. And of course, for both systems, you will have an equilibrium distribution, right? Your Boltzmann distribution that is given by, it's proportional, let's say, to the exponential of the Hamiltonian describing your system. What we would like to have for the coarse-grained system is an Hamiltonian that essentially gives us a probability distribution that is consistent with the atomistic one, with the high resolution one. And what does it mean? So intuitively, we can think about consistent in this way. So this is a free energy surface. I guess you all now know what a free energy surface is. It's up to constant minus logarithm of the probability distribution. So you can think about it as a probability distribution. For a 2D system, it's a toy system. And if we want to coarse-grain this system to a single dimension, so we only take the X coordinate, we have to marginalize this probability distribution. So the free energy will look something like this. This is what we expect, right? This can be extended in terms of multidimensional problem. And we know how the coarse-grained potential is. We have an equation that tells us exactly the solution for this coarse-grained problem from a bottom-up perspective. The problem is that this integral is not solvable for anything that is not a toy system, because this integral runs over the entire phase space. So we want to, since we want to solve a real system, this integral runs over millions or billion of degrees of freedom. So it's not tractable in practice. This is why in 1994, Jacopo Venturin reformulated this coarse-grained problem. So instead of solving this very high dimensional integral that it's not possible, you can obtain at the same way the coarse-grained potential as the absolute minimum of a functional, the so-called force matching functional that Gianni mentioned before. Don't want to go too much on the details regarding this function. If you're interested, we can discuss about it later. But basically, the idea is that you are measuring the difference in the forces generated by your 3LCG potential, if you want, that are given by the gradient of your coarse-grained energy, and the atomistic forces mapped onto some coarse-grained representation by some operator that you define given the coordinate mapping operator. Theoretically, we know that the absolute minimum of this functional is the exact coarse-grained potential, the exact potential of mean force, as is also called. The problem, of course, is that this is exact in the limit of that you can span the entire space of functions. So this U has to be expressive enough in order to span the entire space of function. And of course, you also in principle need to have an infinite sampling regime. We don't have any of those things. First, we have a finite sampling regime. So we need to represent this loss function, as you might guess, considering the fact that we have a finite number of samples from our reference distribution. And typically, this method was used using some basis set, right? So you have some guess for your function, for your coarse-grained force field. But it turns out that this method was not applicable using fixed functional form. We need something more expressive. And this is why we started to use machine learning. Also, this is also what Janis said before. We had lots of collaboration with the group. What I want to say here is that we mainly use graph neural network for doing that for several reasons. One main reason because they are extremely flexible and by construction transferable for different configurations. And another thing I want to say is that we found that the introduction of some prior knowledge that we have for our system is fundamental in order to stabilize the prediction. You can think about those prior terms as some pre-knowledge we have about the system, right? So for example, the fact that two beats shouldn't overlap, or two beats that are topologically bonded shouldn't go too far away, right? So we add some terms in order to enforce all of this knowledge we have about the system. And this makes the simulation much more stable when we end up to simulate this model. And the very first result I want to show you is for a protein specific. So here you're looking at, again, the free energy surface for a very small molecule that is chignolin as a function of two reaction coordinates. We discussed before about reaction coordinate. I don't want to go too much into the details, but basically, those are Teague's reaction coordinates, if you're familiar with them. Basically, they describe this lowest motion inside our system. And for this reference, atomistic reference, we need to simulate something around 2,000 atoms explicitly, so all the protein and all the solvent. And we use Markov state model in order to get the result, because direct molecular dynamics, it's already for a system that is very small like this, not feasible in a reasonable amount of time if you want to obtain the entire free energy surface. While for the coarse-grained model, we only need to simulate 10 beats. So the reduction is evident. We go from something around 2,000 to something around 10. Because in this case, we only retain one coarse-grained beat for every amino acid. And just to show how good are those results, this is also the 1D projection, so you can really see how we can match the atomistic result. The problem with this first plot I'm showing you is that this is a protein-specific model. It means you already have your reference data and you train a machine learning model or whatever to reproduce exactly those data set, right? So it's not extremely useful. It can be kind of useful, but not extremely useful. What we would like to do is extrapolation at the end of the day. We would like to train this model on a subset of protein or something and make prediction ideally on everything else. For doing that, we need to do what Gianni showed before, right? We need to be sure that this model is able to interpolate. So we need to be sure that this model, if trained on several different targets, several different proteins, is able to recover the same result that you would get training a protein-specific model. And this is exactly what Gianni presented before. So with this paper, we demonstrated essentially that we are able to interpolate, to make interpolation. But in order to be more general, what we want to do is something like this. So we have this pipeline in order to train a more general models. So we have to start with some very diverse data set. We coarse grain it to some specific resolution. In the case of the work I will show you, it's 5-bit resolution. It means for every residue, we keep 5 coarse grain bits that are the heavy atoms and 1 carbon beta corresponding to the side chain effect, if you want. Then we, of course, have to design the prior model. And this is it's very complicated to do that. It turns out to be one of the most complicated part when you want to build a transferable coarse-grained force field. Then we can train our CG model to correct for the to take correction for the prior prediction. Okay, and for that we want to test the model using systems that were never seen during training. So the data set that we design, it's made of 50 different structures from the CAF domain, simulated around their folded state. There were something like in between 50 and 70 residue length. We also added some dimers and monomer interaction, especially couples of dimer that were simulated very close and very far apart using umbrella sampling. This was done in order to, let's say, give the model some information about long-range interaction and very short-range interaction. And as a validation set, we use different proteins that were never be seen during the training set. So they are completely outside of the training set. In particular, I want to point out the fact that, especially for bigger system, the sequence overlap with the everything that was inside the training dataset, it's very, very low. Of course, the smaller system has higher sequence similarity just because it's more probable you see something like that because they are very small. And this is the first result I want to show you. So here we are comparing the free energy surface we can obtain with the reference free energy surface that we were able to simulate. So those systems are quite small, so we can somehow still recover the free energy surface. And on the top row, there's the simulation for 2 octapeptides. So they are not really proteins, but something smaller. And then we have results for signaling, tripcage, VBA, and vilin that are the smallest protein you can think about if you want, that are really proper proteins. In this case, again, we are able to reproduce the folded state for most of the system, and especially we are able to reproduce most of the metastable state. This is the fundamental part. We are not just interested in reproducing the folded state. We could use AlphaFold or something else for that. We really want to reproduce the entire free energy surface, meaning the entire probability distribution for our system. And the most impressive part is that if you compare the result that we obtain with other state-of-the-art coarse-grained force field, we can do much better. Here, for example, it's a comparison with Awesome and Unrest that are 2 other state-of-the-art non-machine learned coarse-grained force field. And as you can see, we are really able to, in most cases, get the folded state correct, and especially to explore lots of metastable state, things that Unrest and Awesome in most cases are not able to do. It has to be said that Awesome, in the case of signaling, for example, is very good to reproduce the folded state, and this will come also later. But this is because Awesome has a memory fragmenter inside its functional form that bias the prediction towards the folded state. So This is why also it's very good sometimes to reproduce folded state for different systems. This is a result for a bigger target, alpha 3D, where we started a simulation from a completely artificially elongated chain, and we let the model run. And as you can see, we are able to recover the secondary structure and the tertiary structure almost perfectly in this case, and we have lots of fluctuation around the tertiary structure. In general, for biggest target like alpha 3D or domain ubiquity, we're not able to obtain a reference free energy surface. So the only thing we can do is to compare, for example, the fluctuation around the folded state. So here you're looking at the root mean square fluctuation around the folded state, and we are generally able to reproduce the shape of this fluctuation correct. Also, if we have more flexibility, and this is something expected, right? Because the coarse-graining procedure make the free energy smoother in a sense. So we expect to have more flexibility compared to the atomistic reference. This is another result where we are simulating proteins between 80 and 100 residues, so much bigger compared to everything we had in a training data set. And as you can see, we are still able to recover the folded state this, but also to explore some metastable state outside of the folded state. So we are really able to interestingly, we are also able to make prediction for IDPs. In particular here, we are looking at the secondary structure distribution in comparison with the NMR distribution. NMR, it's an experimental set that we have. And also in comparison with the AlphaFold prediction. Again, we are more flexible compared to those results, but this is somehow expected. What I want to point out here is that if we look at the free energy surface that we obtain from our simulation, we can, of course, stabilize states corresponding to the NMR distribution. So the minima of the free energy that we get correspond exactly to what we would expect from the NMR. But we also get some other different state, like this state 4. It's a metastable state completely outside of the main basin. And just by looking at it, you might think this is garbage. It's something that the model predict that is simply not correct. We also thought that at the very beginning, but it turns out that it's not garbage. It's something that it's really a metastable state in the force field we are using to train our model. So if you run very, very long molecular dynamic, you can see transition from this basin here to this other basin here. And this turns out to be a stable basin, both in the force field we use, that is amber, and in another force field that is char. So if you run simulation using different force field, this is a real metastable state that you probably will never find if you just run molecular dynamics, because all your reference sample are distributed in this main basin here. So why you would like to explore something more at the very expensive cost of molecular dynamics. With that, we can also look at some more quantitative metric in order to quantify the ability of our model to reproduce the folded state ensemble, in particular the GDTS score that was using CASP or the largest metastable fraction of native contact. And in general, you can see that we are better than all the other state-of-the-art force field. We are sometimes even comparable with DESRES prediction. DESRES is a atomistic force field, so a standard molecular dynamic force field that we use here to compare our prediction with something that is really high resolution. DESRES, of course, was never used during the training of our model. Again, also here, the ability of ozone to reproduce the folded state seems to be better, but this is signaling just to highlight it. But the point is, again, that ozone was biased in order to reproduce the folded state of signaling. So it's somehow expected that ozone is able to fold very well this protein. But as I said before, folding is not the full picture for this kind of model. We really want the entire probability distribution, the entire free energy surface. So those metrics are important, but they do not tell the full story. Okay, so we have a very fancy model that is able to make extrapolation, so to predict some result for system, but we don't want it to be just a black box, right? We want to be sure this model is predicting something correct. And this is why we started a collaboration with Klaus Robert Müller Group in TU Berlin in order to apply GNN-LRP in particular to our model. GNN-LRP is an interpretation technique that basically decomposes the network output for a graph neural network in relevance that is associated to several walk of the graph. So you can think about it as a sort of multi-body decomposition, since our output is the energy. And from that, we can get some insights. So we can see if the prediction from a relevance point of view are consistent with what we would expect from a physical point of view. So for that, we tested two different graph neural network that are PAIN and SO3net, very different graph neural network for two different systems, bulk water and bulk methane, both coarse-grained to retain only the central heavy atom. First, both models are able to reproduce correctly the RDF in this case. Of course, the RDF for water is much more peaked in this region, while for methane, it's more like the one that you would get from a Lennard-Jones fluid. In general, methane, it's very, very simple from a coarse-grained point of view. While water, since it has very complex hydrogen bonding structure, it's much more complicated to treat. Also using graph neural network, you cannot use something like SNET, for example, to obtain a good result for water. And then we overimpose here the relevance curve for the two-body relevance. So we collect all the two edge walk inside our graph, and we compute this relevance. And as you can see, we have something that is reasonable. So we have stabilizing energy contribution from the two-body information that correspond exactly to the peak for water. And we have this repulsion that we can observe both in water and methane that is associated to the, let's say, low distance between two beats, right? So it's again something that we would expect. And we also expect the fact that the relevance goes to zero as soon as the distance increase, right? Those curves are at a different length because of the network cutoff. So the network cutoff is different, and this is the reason. For methane, the minimum, it's very it's not so pronounced, and this again indicate that methane doesn't have lots of information from the coarse-grained point of view. This is also the reason why you can obtain a good coarse-grained model for methane without any neural network behind. So it's much easier compared to water. We can also look at some free body information. So this is the distribution of the angle that we have between three different molecules. And for water, again, for example, this first peak corresponds to the tetrahedral structure of water, so the tetrahedral arrangement. And again, the relevance has a minimum here in both model, while for methane, we have a completely flat completely flat relevance curve. And this really indicates, again, the fact that methane doesn't have multi-body information. So we don't need multi-body information for realizing a good coarse-grained model of methane. This is, again, in agreement with the fact that if you build a coarse-grained model just using two-body information for methane, it works very well. While for water, it just don't work. It's not possible to let it work for water. We can use this interpretation method also for proteins. So in particular here, I'm showing you the free energy surface and draw of NTL9. Also, Jan, you talked about NTL9 before. It's an interesting system because it has a mixed secondary structure. So we have alpha structure and beta structure. And the reference free energy surface present a folded state, an unfolded state, and two measure folding pathway. So we have this P1 folding pathway where we have beta 1, 3, and the alpha helix already formed, and the protein folds by forming beta 1, 2. And P2, where beta 1, 2 is already formed, and the proteins fold by forming the alpha and the other beta region. RCG model, that in this case was based on pain, is able again to reproduce folded state, unfolded state at the two measure folding pathway. The idea here is then to apply again this relevance method in order to understand our model better. So just focus on the left plot. Here we are looking at the absolute relevance that we get from the two body contribution in the folded state and in the unfolded state on the bottom. So as we would expect in the folded state, most of the relevance is located where we have the secondary structure formation. So in the two beta regions and in the alpha region. And most of the contribution are stabilizing the interaction. While for the unfolded state, we have a more spread information around all the system, and most of the contribution are destabilizing the structure. So this is again something that we expect. On the right, instead, we are looking at the relevance difference between structure in the folded state and structure in P1 or P2 state. What does it mean? It means that if this is red, basically this state is less, this relevance information indicate that this is less stable from the energetical point of view compared to the folded state, while if you see something white, essentially this is as stable as in a folded state. And again, the results that we get are completely in agreement with what we would expect from the knowledge of our system. So in P1 state, this tells us that the alpha region and beta 1, 3 are already formed, and beta 1, 2 is not as stable as in the folded state. So again, this is completely correct from the physical point of view, and the same for P2. So beta 1, 2 is already as stable as in the folded state, while beta 1, 3 and the alpha region still has to be have to be formed. But with this relevance method, we can also study mutation, and we can get some information about mutation without need re-simulate or test anything else. We can just change the beta identity inside our sequence. Mutation in coarse grain, it's very, very simple because you, especially when you have a C alpha model, you can just change the identity of your bit and make a prediction, right? And this is exactly what we are doing. So on the top here, we're mutating this leucine 30 with a phenylalanine 30. So the leucine 30 is known experimentally to form very stable interaction with this leucine 35 here, and in general to stabilize all the alpha region. And if we mutate it with a phenylalanine that is much bigger, we expect some destabilization in the interaction, of course, with the leucine 35, but all over the alpha region in general. And this is exactly what we can see. So this point here corresponds to the interaction with the leucine 35 that, as you can see, it's destabilized after the mutation from the relevance point of view. And in general, we have destabilization spread all over the alpha region. The same if we look at this isoleucine for aspargine mutation. So we are mutating something that is hydrophobic with something that is hydrophilic. And in this case, we expect, again, a destabilization of the entire beta region. And as you can see, this is again, exactly what we get from the relevance point of view. So this really indicates that our model is learning the correct physics that we would expect from our system. Okay, so With that, I want to move to the next topic, that is how we can include nuclear quantum effect. Why I'm talking about nuclear quantum effect here? Well, because one way to incorporate nuclear quantum effect in molecular dynamics is to use Feynman path integral formalism, and in particular the so-called ring polymer model. And this will involve then another kind of coarse graining. So we are not coarse graining a protein, but we will coarse grain this ring polymer. First, let me start by introducing nuclear quantum effect, what they are and why do we need them. So in general, as I said before, molecular dynamic simulation relies on the Born-Oppenheimer approximation, right? So we can separate the electronic degrees of freedom and the nuclei degrees of freedom, and we treat the nuclei normally as classical particles that moves inside the electronic field. The problem is that in principle, nuclei are not classical particles. So nuclei also obey the Schrödinger equation. What does it mean? It means that there are some regimes where we cannot ignore the quantum effect of nuclei. For example, in the case of very light nuclei, the quantum properties emerge very clearly from the system, or also if we go to very low temperature where lots of experiments are done. So in those cases, we need to explicitly take into account nuclear quantum effect. And as I was saying before, this can be done considering the Feynman formulation of quantum mechanics. So basically, the main idea, the classical picture that is always told is that if you want to move a quantum system from a point in phase space A to a point in phase space B, it doesn't just follow a single path, right? It follows all the possible classical paths at the same time, of course, all weighted by some quantities that is dependent on the entropy of the system, but don't want to go into that. The point is that if we discretize the Hamiltonian associated to this quantum picture, we obtain something that is exactly the same of a classical Hamiltonian of your system, but instead of just one copy of your system, you have P different replicas of the same system, and all are connected to the next one by a spring potential, so by an harmonic potential. This is the so-called ring polymer. The theory is correct in the limit of number of replicas going to infinity, but in practice, this number of replicas, P, is more like a convergence parameter. So we need to increase it as soon as until the simulation converge. One of the major problems here is that these number of replicas has to increase when the temperature decreases. So of course, as expected, right, where the quantum effect became more relevant, we need to add more replicas to the ring polymer in order to properly describe the quantum effect. And this is, again, something that is known for from a long time. And this idea of coarse grain the ring polymer is not something completely new. There were some attempts of doing that before machine learning, but basically nothing worked also in this case because they just use fixed functional form. Our idea, again, was to use a machine learning network. In particular, we use Maze in this case, to apply the coarse grain technique to the ring polymer. So we coarse grain the entire ring polymer to just a single replica. So we basically recover the entire quantum description at the cost of a classical molecular dynamics simulation. And there are two different route we can take. One is for quantum statistic and the other is for quantum dynamic. In the case of quantum statistic, we coarse grain the entire system to a single replica in the ring polymer. And those are some results that we have. So for example, in the case of a water molecule, in blue is the prediction that you have considering a classical molecular dynamic prediction, so a classical molecular dynamic distribution. And in yellow is the correct quantum prediction that you have simulating the entire ring polymer. And in black is what you would get considering our method. So in this case, the cost is the same of the blue prediction, but the result is perfect for different distribution and also different temperature. Also more challenging system like Zunder-Cation that are two water molecules with a nitrogen in the middle. In this case, again, we can perfectly recover the quantum distribution at the cost of the molecular dynamics simulation that is completely off, especially when we go to low temperature. We can also recover the correct result for bark water, so in the case of a more extended system. What is interesting is that since the temperature dependency and the mass dependency in the spring force is trivial, we can also recycle essentially data that we obtain at one specific temperature for one isotope composition to train a model to another temperature and another isotope composition. This is useful, especially because generating data at high temperature, I said it's cheaper, right? Because you need to simulate less replica. And by reweighting this distribution, we can really get the same result that you would get simulating the bigger ring polymer at different temperature, for example. And this is for an example of result that we can get. In the case of quantum dynamics prediction, here we need to coarse grain the system to the centroid of the ring polymer. And this is because from the theory, we know that in order to recover properties such as the infrared spectrum, we need to look at the centroid evolution. So we need to compute correlation function centroid distribution. And in standard molecular dynamics, in standard path integral centroid molecular dynamic, they have to simulate the entire ring polymer and take step by step the centroid. In our case, again, we can coarse-grain the entire system directly to the centroid and learn the effect of the ring polymer. Again, obtain results at the cost of a classical molecular dynamics simulation. One major problem of this centroid molecular dynamic is the so-called redshift that we have as soon as we go to low temperature. So this is something well known. But since the temperature dependency of the correlation function in the zero energy state, let's say in the ground state of our quantum system is trivial. So we can essentially take a correction for this effect by just reweighting our simulation essentially. So the idea is that we can use a temperature elevation ansatz that basically work in this way. So we train our model to a high temperature that has to be high enough in order for the system to be in the ground state, but again, high enough that we don't have this redshift effect. Train this potential, we can then use it to make simulation at lower temperature and obtain the correct result that also doesn't take into account the redshift effect. And this is an example of what we can get. So this is just a toy system, so Morse potential that mimic the OH bond. And this is the exact solution that we get for the infrared spectrum at different temperature. And this is what you would get from the centroid molecular dynamics simulation. So again, here you need to simulate the entire ring polymer, and your prediction is not very good because you have this redshift effect that I was mentioned before. In our case, instead, that is the red prediction, we can, we can cheaply compute this spectra and correct for the redshift effect. The same story, basically, if we look to something more realistic, like water molecule distribution. So in the case of a water molecule, we can still recover the correct spectrum, essentially, and correct for the redshift effect. And as it was mentioned, this becomes more and more efficient as soon as we move to low temperature. We can also reach cryogenic temperature, where some experiments are perform normally, just by using this temperature elevation ansatz and still recovering the correct distribution at the cost of a standard molecular dynamic simulation. The same also if we look at bulk water and hexagonalized results, we can still get the correct peak for the infrared spectrum and also Raman as anisotropic information. Basically, again, the same result if we look at the water-air interface or Zunder cation at very, very low temperature or water dimer. This method here in particular, this gray MCTDH method, is a solution on the fly of the time-dependent Schrödinger equation. So this is very close to the experimental value, but consider this is not feasible for anything but Zunder cation maybe, or not something bigger for sure. While with our method, we are as expensive as the molecular dynamic curve, that is the blue curve here, but we can obtain the correct result. And so with that, we concluded the bottom-up perspectives, because also in the case of this path integral formalism, we use bottom-up approach in a sense, and we move to the top-down perspective. So we want to use to understand how important are multi-body terms for a coarse-graining model in order to reproduce some. In particular, here we will focus mainly on delta G prediction. So in order to do that, we use this so-called ODEM method, that is observable driven design of effective molecular models. Basically, the idea is that we want to match experimental observable, right? So let's assume we have an initial function that describes our coarse-grained potential that will depend on a set of parameters, right? And we select some observable we want to optimize our coarse-grained potential for. So we have our set of observable F, and we initialize our model with an initial guess, let's say for the parameter. This guess can be really a guess, or it can be something that we know from other model, like, I don't know, bottom-up perspective, for example. So we train a model from a bottom-up perspective. We have a set of parameters. We initialize this model, and we want to optimize this model in order to reproduce experimental observable. What we can do then is to simulate this model and apply perturbation theory. So we perturb the equilibrium distribution that we get using this initial parameter guess, and we compute perturb value of the target observable we want to optimize the model for. Yeah, with that we can compute the likelihood between the experimental observable and the target observable that we obtain from our simulation. And by optimizing this likelihood, that is essentially a loss function, we can obtain a new set of parameter epsilon prime. Unfortunately, this set of parameter cannot be the initial set, because otherwise the perturbation method just falls down. It needs to be quite close in order to use this perturbation approach. So we need to iterate this method until we reach some sort of convergency. We use as target observable delta G prediction, the difference in free energy between folded and unfolded state change upon mutation. And we found this is a good candidate for this method, because for coarse graining, again, compute delta G, it's very cheap. You can use, again, perturbation theory, while using atomistic representation, it's more complicated. You need to struggle a bit. The two targets that we use were ubiquitin and protein G, both coarse-grained at the carbon alpha resolution. So again, we only retain a single coarse-grained bead for every residue. And what we optimize was two-body interaction only, and then multi-body interaction in order to see how the inclusion of multi-body interaction improved the model prediction. In this case, the functional form that was used for the coarse-grained model is a fixed functional form, so no neural network here. And for ubiquitin, you can see that the addition of many body information, it's not very helpful, let's say, to improve the model prediction. But for protein G, the multi-body terms are necessary in order to improve this kind of prediction. So here we really need multi-body terms. Of course, again, these results are not extremely good, let's say, but this is because we use only a fixed functional form for our coarse-grained potential. So we are now working on the application also here of a neural network potential in order to improve these results and to make them more useful. So with that, I want to conclude saying that at least from our group point of view, the final objective of coarse-grained modeling would be to have to combine somehow the top-down and the bottom-up perspective in order to obtain some model that is as general as an all-atom force field, extremely cheap in order to make simulations that are reliable and that can be used also for systems that are not just toy systems. And with that, I want to thank you for your attention. I want to thank all the members in Cecilia Clementi's group. I want to thank also Cecilia for all the work she has done and she's doing for leading all of this. And I'm open to any question. Thank you.
All right, if you have a question, just raise your hand and I can bring the microphone to you. Okay.
Thank you for your talk. Could you clarify when you said that it wasn't working for the water molecules, but it was working for methane? Can you go back to that slide just to clarify it again?
No, the results I'm showing here are both working for water molecule. The point is, if you use some different network that only take two-body information, like GNet, for example, you cannot reproduce the RDF for water. Also, if you use something like general two-body potential with a fixed functional form, you cannot reproduce well.
RDF here.
Sorry?
What do you mean by RDF here?
The RDF is the radial distribution function. So it's a two-body quantity that we can make. Indicates essentially the density that you, in probabilistic term, the density that you have given a specific distance from your target atom, if you want.
And I'm trying to understand what is the takeaway message from this slide? It still wasn't clear.
The takeaway message is that the relevance is somehow a way to understand the model prediction, right? So we can simulate our model and make prediction, but we cannot really say that the prediction that they are doing from the energetic point of view is correct for every system, right? It's correct as a mean, so in terms of distribution, but we want also to be sure that this is consistent with the energetic prediction. Okay, so these relevance methods allow us to say, well, where we see in this RDF that is a sort of, you can think about it as a transformation of the probability distribution, if you want, where we have a peak, meaning we have high probability to find a molecule there. It means this is stable from the energetic point of view. And this is exactly what this relevance curve tells us. So this minimum here indicates that from the energetic point of view, in terms of two-body interaction, we have a stable point here. And this corresponds to the peak of the RDF. That is like, again, a peak of some sort of probability distribution, if you want. Again, for example, the same for the fact that we have this repulsive from the energetic point of view, right? Because I said methane behaves more or less like a Lennard-Jones fluid, so you would expect some sort of repulsive core there. And what this is telling us is that this repulsive core actually exists. So it's an indication that our prediction is correct. And the model is learning something that makes sense from a physical point of view, because we really want our model to be meaningful from a physical point of view. Yeah. Oh, sorry.
I have a question. What is the scale? How far could you push in terms of coarseness your approach? Like how many molecules or atoms or whatever? That would make sense.
Yeah. So it really depends on the system you want to study. So for example, for proteins, it doesn't make sense to go below C alpha resolution, I would say, if you want to preserve some indication of the secondary and tertiary structure of the protein, right? You cannot go below 1 bead for residue. You could in principle, but don't know if it makes much sense, right? The specific resolution you need to use in order to have a good model, it's another story. So until now, we're not able to obtain a good transferable model at the C alpha resolution. So we need to use 5-bit resolution because it turns out to be easier in terms of building a transferable model. But consider again, this really simplify a lot the problem, because you eliminate all the solvent information that is completely eliminated. And for every residue, you only take 1 or 5 bit. So it's reducing a lot the number of degrees of freedom you have. And of course, for a different system like bulk water or bulk methane, it's different. There, the coarse grain is less aggressive because, of course, doesn't make sense again to go beyond something like this. So in the case of water, we only eliminate 2 atoms, or for methane 4, but for every molecule. In the case of proteins, again, we normally work at C alpha resolution, so 1 bead for residue, or 5-bit resolution, means 5 bead for residue.
Perfect. That's all the time we have for questions today. So thank you again to Jacopo for the great talk.
Thanks.
Let's give a round of applause.
Thank you.
And we'll jump